but in the past, we had a scholarship program, the Rick Iverson Scholarship Program, whereupon we would award a scholarship to an emerging student who was working in the field of invasive species. And so our last award was given back in 2019, and we awarded that to Christina Mitchell, who is now here today as part of our tradition to discuss the work that kind of won her that award and what she's been doing since then. And I, for one, am really excited. This will be the second talk today that delves into the underworld of invasive species. You know, we looked at the seed bank earlier, and now we're going to explore, you know, the effects of invasive plants on forest structure and arthropod communities in the forest. So this is a very interesting ecological dive. So I hope you all enjoy it. And now, Christina, I'll give you the floor.
Thank you for that introduction. Just verify that the screen is correct.
Perfect.
Thank you. So yeah, today I will summarize results from my PhD research that highlights the effects of non-native plant invasion on urban forest communities. So we know forests are important ecosystems. However, most of the forests across the United States has been cut down, often for agriculture and development, and these are both practices that still occur globally. Remaining forest patches provide valuable resources like atmospheric cooling and native species habitat. Over 4 billion people live on less than 3% of the Earth's surface, which means cities have a lot of impenetrable infrastructure on a small area of land. Therefore, forests are threatened by development as urban areas expand to accommodate growing urban populations. Remaining forest patches can be negatively affected by urban pollution, the urban heat island effect, which describes the occurrence of cities being a few degrees warmer than surrounding areas, and invasive plant and arthropod species. The picture on the right shows non-native English ivy growing from the ground layer up the tree trunks, and this can eventually lead to canopy dieback and tree death. When I say urban forests, I'm referring to relatively unmanaged patches of forests around urban centers. And I say relatively because some of these forests have walking trails that may occasionally get cleared, but the forests besides that are unmanaged and a canopy persists despite the dirt path. These urban forests can be surrounded by impervious surfaces like buildings, roads, and houses, and other vegetation types like turf and agricultural crops. Interest in urban ecology research has been growing during the last 30 years, but many aspects of urban forest ecology are still unknown. Of particular interest is the potential for urban forests to provide insight on how non-urban forest communities may respond to future warmer climate conditions and increased occurrences of non-native species invasions. Previous research comparing urban forests to rural forests has found that urban forests are usually smaller. This means they tend to have a greater ratio of forest edge compared to forest interior. Additionally, urban forests tend to have a more open canopy and a denser understory, which is often invaded by non-native plants. Urban forests also tend to have less litter debris, including leaves and coarse woody material, and the litter and soil layers tend to be drier. There's also been research on urban animal communities that shows high variability in community composition between different cities and between taxa. However, cities tend to host more generalist species and species that specialize on vegetation types without canopy, including grasslands, shrublands, and open ground. Most studies do not relate multiple taxa to each other, so we quantified plant and arthropod communities in urban forests to understand how changes in plant communities and related changes in vegetation structure affect arthropod community composition. In particular, we were interested in the effects of non-native plant invasion, a common occurrence in urban forests, on forest structure and arthropod communities. We used 24 forest research sites from the FRAME network. FRAME stands for Forests Among Managed Ecosystems, and this long-term research project allows researchers access to a network of over 50 forests and the associated data. We sampled plant and arthropod communities in 12 forest sites around Raleigh, as well as 12 forest sites around Newark, Delaware. And the reason for this was to compare communities between different regions and different species pools to determine whether urban communities respond similarly to effects of urbanization. As an example, this map shows the distribution of sites across Raleigh. All forests were selected to establish a range of sizes and levels of urbanization, and they were selected without prior knowledge of its site conditions, so the internal characteristics. For all 24 forest sites, we either sampled the entire forest or a portion of the forest. We sampled a portion of the forest in about half our sites in situations where the forest patch was too large or we did not have permission to sample the entire patch. And then within the sampled area, we established a 25 meter by 25 meter flagged grid, which is represented by the white dots in the diagram on the left. And we randomly selected 10 points for data collection, which are represented by the black dots. And all of these data collection points were over 25 meters apart from each other. One of our 24 forests only accommodated 9 points because it was pretty small. So we therefore collected all data that I will refer to from these 239 sampling points. And so I'll now briefly cover how we collected the vegetation data because I used the same vegetation data for the analyses that you'll see. So first we quantified understory density using what's called a NUDS board. The picture on the right shows some extreme examples for each city. And for reference, that black and white NUDS board, it's 2 meters tall, and it's in the same location in all 4 of those images. It's just obscured by vegetation in the bottom 2. We also quantified canopy openness using a densiometer. So the image on the left shows an example of how canopy cover is displayed in this instrument. And within our sampling area, we estimated percent cover of total ground layer vegetation, including percent cover of non-native and native ground layer vegetation. So this picture on the right is showing some English ivy and other species growing along the ground. That's what we would have measured in this instance. We also counted and identified all plant stems to determine the density of non-native and native stems in the shrub layer. And so the diagram on the right is demonstrating how single plants can have multiple stems. And lastly, we quantified leaf litter volume by measuring a sample from each point. So the work I'll outline in the rest of this talk was conducted in these 24 urban forest sites at the same sampling points where the vegetation data were collected. From our understanding of urban forests and how non-native plants affect forest structure, we first sampled ground layer arthropods and focused on carabids, as they are a known ecological indicator of forest condition. We then moved up to understory arthropods and wanted to understand community composition more broadly, so we focused on all collected arthropods. So we know urban forests can harbor non-native species, which can alter forest understory structure. We used carabid or ground beetle species as a model community because they are taxonomically and ecologically variable, and they're numerous, which allows for adequate community sampling. Additionally, carabid communities change in response to altered biotic and abiotic conditions, included human-mediated disturbances like agriculture and development. To quantify the differences between community composition, we were interested in 2 groups of carabid specialists. We focused on forest specialists as they represent species evolved to belong in a forest, and open area specialists as they in essence represent species evolved to not belong in a forest. So on the left are 2 forest specialists, and both rely on closed canopies and moist dense litter for survival. I've highlighted the mouth parts of the carabid on the right to illustrate that this species specializes on eating snails and is adapted with a narrowed head and spoon-shaped palps for scooping out that snail meat from the shell. On the right are 2 open area specialists, which is the term I'm using to refer to species that specialize on vegetation types without canopy. So this includes grasslands and shrublands, as well as areas of open ground. And these species specialize on open areas for food resources and dry soil for larval development. And their abundance can increase in response to greater soil disturbance and annual plants. Previous comparisons indicate carabid community composition varies along rural to urban gradients, and often in predictable ways. In study communities, urban forests often host more generalist species and open area species, compared to rural forests that typically host more forest specialists. We hypothesized that this was caused by the effect of non-native plant invasion on changing the structure and microclimates of urban forests, and therefore wanted to compare urban forest crabbed communities along a gradient of non-native plant invasion. So if we return to the general setup of our sites, again, we collected all data from 239 sampling points, and to quantify the understory vegetation community, we used our previously described canopy openness, understory vegetation density, stem density, ground cover, and leaf litter data. To quantify the crabbed community, we activated pitfall traps during 2-week intervals between May 2017 and July 2018. So we sampled crabbids 8 times over 14 months. And trap deployment or crabbid collection was typically done for all forests within the same week. So I would drive first collect, deploy or collect all the traps from Raleigh and then in Newark. And I then identified crabbids to species and categorized each species as a forest specialist, an open area specialist, or neither. So here's some summaries to give you a sense of the communities I'll be describing. From these 24 urban forests, we identified over 100 plant species, including over 30 non-native species. We captured over 1,800 crabbids, representing 57 different species, and we captured more forest specialists in Newark and more open area specialists in Raleigh. We used a multivariate technique called non-metric multidimensional scaling to assess community structure and similarity. In brief, the closer together the forests, which are represented by the letter combinations and color-coded by city, the more similar the corresponding crabbed communities. And I won't go into detail, but the black arrows represent certain vegetation features that influence the differences between urban crabbed communities. Together, this information demonstrates that crabbed communities differ between Raleigh and Newark forests and are affected by different aspects of understory vegetation. Thus, we chose to analyze our cities separately and focus on understory vegetation as a mechanism for crabbed community change. We then used structural equation models, or SEMs, to find support for our a priori hypotheses. First, that canopy openness increases understory vegetation. Second, that non-native and native understory vegetation affect forest understory structure and leaf litter. And third, that all these things directly and indirectly affect crabbed communities by reducing forest specialists and increasing open area specialists. But instead of showing you 12 similar and intuitive looking models, I'm going to walk you through the results in a simplified table form. The crabbed response variables are along the left side of the table, and the predictor variables are along the top of the table. In general, canopy openness increased understory vegetation density, especially non-native plants, which had different effects in each city. For reference, significant relationships in Raleigh are shown with solid orange arrows, and Newark relationships are shown in purple arrows with white dots. Native ground cover increased forest specialists in Raleigh, and non-native ground cover increased open area specialists in Newark. However, native vegetation also decreased forest specialists in Newark. Canopy openness increased open area specialists in Raleigh. Understory vegetation density indirectly reduced open area specialists in Newark. And leaf litter volume both decreased open area specialists in Raleigh and increased open area specialists in Newark. We believe the city differences between forest specialists and native vegetation and open area specialists and leaf litter volume are caused by the underlying difference in each city's plant community. Raleigh had more non-native understory vegetation, and Newark had more native understory vegetation. We believe this difference influenced the relationships we documented between non-native and native plants and crabbed specialists. However, both types of plants contribute to dense understories, which generally decrease crabbids. Regardless of whether non-native or native plants were more common, canopy openness increased non-native plant invasion, and non-native plant invasion in turn reduced forest specialists and increased open area specialists. We also found evidence of competition between non-native and native ground layer vegetation, suggesting non-native plant invasion may inhibit native tree regeneration and prevent long-term canopy cover in urban forests. Some of our previous results show that while all crabids increased with greater forest size, small urban forests still had comparable forest specialist communities to some large forests. Together, we recommend the removal of dense non-native vegetation, particularly in the ground layer, to allow unsuppressed development of the native canopy for maintained canopy cover to benefit forest specialist species. Additionally, we recommend conservation efforts target urban forests with a focus on preserving intact canopies, rather than a focus on size. Our results indicate canopy openness increased non-native plant invasion, so prioritizing canopy closure in the short term can help reduce non-native plant invasion and associated management costs in the long term. Our results extend past understanding urban forest crabid communities. Predaceous crabids can control forest arthropod pests that may increase with warming climate conditions, and conserving forest specialist species habitat also conserves resources for other organisms, and stable forest arthropod populations provide food to sustain bird, herpafauna, and mammal population. We show forest specialist crabids increased in response to native ground cover and decreased in response to non-native ground cover. It's not a far stretch to think other forest specialists may be similarly affected by non-native plants and understory density. So now we understand a bit more about how understory vegetation and non-native plants affect crabids, which are ground-dwelling arthropods. But now we want to investigate how understory vegetation and non-native plants affect understory arthropods. Ecological theory and empirical evidence indicate greater structural complexity and diversity in plant communities will increase arthropod abundance and diversity, while non-native plants are typically associated with low arthropod abundance and diversity. However, we know non-native plants can increase understory vegetation complexity and that non-native plants are common in urban forests. Therefore, urban forests are the perfect system to test the question. Will structural complexity associated with non-native plants increase abundance and diversity of arthropods, as predicted by plant complexity literature, or will structural complexity associated with non-native plants be depauperate of arthropods, as predicted by non-native plant literature? We collected understory arthropods using a leaf blower on the vacuum function, with a metal funnel inverted to prevent the mesh bags used for sampling from getting sucked into the motor. We vacuumed arthropods in each city in June and July of 2019. I vacuumed all vegetation I could reach with the leaf blowers up to 2.3 meters high in roughly an 80 meters squared area. And this sampling area was centered around each of our 239 sampling points. I vacuumed arthropods for 3 minutes per point, and arthropod samples were then carefully processed to remove all non-arthropod material, and non-spider arthropods and spiders were placed in separate pre-weighed vials. We dried understory arthropods first under a hood and then in a drying oven, and finally weighed them in milligrams. For analyses, we quantified abundance, biomass, richness, and diversity for all arthropods. In analyses, I've grouped these into non-spider arthropods, which I will from here on refer to as arthropods, and spiders. Based on our interest in the effects of non-native plant invasion, we wanted to know whether non-native or native vegetation affect total structural complexity differently. We used frame, ground cover, and stem density data to create 3 structural complexity metrics to represent total non-native and native understory vegetation. Briefly, we determined the full range of data for total ground cover and total stem density, and assigned 5 bins, labeled 1 through 5, to equally span each variable's range and represent increasing structural complexity in each vegetation layer. To create each structural complexity metric, true data values for total non-native and native vegetation were attributed to bins. Bin numbers corresponding to ground cover and stem density were added together to quantify understory structural complexity for each sampling location. First, I'll summarize the arthropod communities. We collected more arthropods in Newark and collected more spiders in Raleigh. Though it was similar between cities, we collected one more arthropod order and two more spider families in Raleigh and Newark. And for those who aren't familiar with insect orders, the most abundant orders that we found were Diptera, which are the flies, the spiders, the Hemiptera, which is kind of the plant hoppers and true bugs, and Hymenoptera, which are the ants, bees, wasps, and sawflies. We use linear regressions to compare relationships between arthropod and spider abundance, biomass, richness, and diversity. For the following figures, trend lines are included in all instances, while 95% confidence intervals, which are represented by the shaded regions, are included to designate significance. And these results show arthropod abundance, biomass, and richness increased with greater total structural complexity, while diversity decreased. If we look at the spider community, these results show spider abundance and biomass increased with greater total structural complexity, but there was no relationship between richness and diversity. We now understand how urban arthropods respond to total structural complexity, but we want to know if arthropods are similarly affected by structural complexity when separated by plant origin. To address this question, we ran models with the non-native structural complexity metric, native structural complexity metric, and the interaction between the two metrics as our predictor variables. I'm graphically presenting the model outputs, and again, trend lines with 95% confidence intervals indicate significance. In Raleigh, non-native and native structural complexity increased biomass. Non-native structural complexity increased abundance, but decreased diversity. In Newark, non-native structural complexity increased biomass, but decreased richness and diversity. Native structural complexity increased abundance and biomass. We found partial support for both the plant complexity hypothesis and the non-native plant hypothesis. Total vegetation and non-native structural complexity both increased arthropod abundance and biomass, but only non-native structural complexity decreased arthropod richness and diversity. This suggests urban forests invaded by non-native plants may still provide adequate food biomass to transfer energy to the next trophic level, for example, from spiders to birds, but may fail to provide unique ecological services and functions required in forests as the number of species filling unique niches declines. Again, our results support the need to consider plant species origin when managing urban green space and to prioritize the removal of non-native structure when allocating vegetation maintenance resources. Let's revisit what we learned today about urban forests and non-native plant invasion. We determined open canopies can increase understory density and especially dense non-native vegetation. We highlighted the effects of total and non-native structural complexity on ground layer and understory arthropod communities. Forest specialists increased with greater native ground cover and forest size. Forest specialists decreased with greater non-native plant invasion and understory vegetation density. Open area specialists increased with greater non-native plant invasion, canopy openness, and forest size. Open area specialists decreased with greater understory vegetation density. Total non-native and native understory structural complexity increased arthropod abundance and biomass, but not diversity. Non-native understory structural complexity decreased richness and diversity. Urban forests are not negligible. They contain diverse plant and arthropod communities and harbor forest specialist species. To maintain urban forests and the benefits they provide humans and other animal species, conservation efforts should primarily focus on preserving intact native canopies over preserving areas of large size. Secondarily, invaded urban forests should be managed for the removal of dense non-native plants. Particular focus should be paid on restoring the native ground layer vegetation, as native tree seedlings may be outcompeted by non-natives and fail to contribute to the future canopy. Finally, we recommend continued monitoring of urban plant and arthropod communities, as changes in urban forests may help inform future changes in non-urban forests. This work could not have been done without every person listed here. These people provided equipment and expertise, field and lab assistance, forest site access permissions, identification verifications, full access to the NC State Rabbit Collection, and funding for my research. I'm very grateful for the North Carolina Invasive Plant Council for awarding me the Iverson Scholarship in 2019 and giving me a chance to present to you today. I would additionally like to thank my PhD committee and the members of the FRAME network. Finally, I thank all of you for your time and attention today, and I look forward to your questions.
Awesome. That was fascinating. Very, very well done. I can tell that you've presented on that often a lot. So that was excellent. It really had my mind racing, thinking about. I grew up in Atlanta and around a lot of urban forests, and I immediately put myself in those parks when I was a kid, and after walking through them in recent years, I realized that the condition is such that you described. It's a mainly native overstory, and almost everything else is non-native. And so the implications for management, I think are important, and also probably presents a little bit of an issue as to how to properly approach that without, detrimental impacts, by removal of too much vegetation.
Right. Yeah, definitely balance.
Definitely. But that was wonderful. I really appreciate it. And we don't have any other specific questions in the Q&A right now, but again, I'll repeat myself as I have over and over. We'll be providing this contact information for these folks. And so anything that your brain thinks about 2 hours from now, like mine does, you'll be able to write it down and hopefully get a question out to these people. So thanks again. And I'd say money very well spent for that scholarship. So
Thank you.
Excellent work. Thank you. Okay, great. Well, we have reached the end of our first day of the symposium. I was thoroughly impressed by all the presentations. I hope everyone else who attended was. A couple things that I'd like to say before we go. First of all, just a couple housekeeping things. Remember the Google form for attendance. Make sure you open the chat up and you fill that out again. I also think most importantly, I want to say thank you on behalf of the Invasive Plant Council for the presenters, for giving us your time and your expertise and your knowledge and keeping this dialogue open. I think it only through collaboration with people like you do groups like us have success. And so it's really important that you're here, and we appreciate it. So thank you. I'd also like to say thanks again to the NCIDT for helping put this thing on and hosting the WebEx platform. Again, we've got another day, and I'll be thanking you again, but things went really smoothly today, and I can't help but think that was because you guys were super prepared. So I want to express my appreciation and gratitude for that. And a couple other things. I want to say tomorrow's session, again, we're starting at 9 o'clock sharp. We will start with a business meeting for the Invasive Plant Council. So for those of you who would like to attend that, please hang in there, and we can talk about board business. We can talk about direction that we're moving. We're also going to be having some important rollovers and elections. And currently, I'd like to announce that we have one position open that will be open. We've got other vacancies filled, I think, but the one position that we will have open is treasurer. And currently, Jonathan Short with the North Carolina Forest Service has been playing that role for the past 2 years. He's done a great job keeping us in line, diligent reporting. And I think if anyone has any questions about what that role looks like, the time investment, feel free to shoot Jonathan an email and ask away, because we will be hoping that we have an eligible candidate step forward at tomorrow's business meeting to fill in that position. So again, Treasury is open. We're going to be having a business meeting tomorrow starting at 9 to 9:45, voting on new membership and officer positions and some rollovers. So I'd encourage everyone to make that meeting. And last but not least, thank you so much, everyone who joined this meeting today in support of us, in support of your own expansion of knowledge regarding invasive exotic plants and the world surrounding them. We really appreciate you. We're here for you and because of you. So please do come back again tomorrow, and we will see you guys in the morning. Thanks so much.